Hello! Scientific method. The scientific method. It's something that you do, something that we do, it's something that everyone does. The scientific method is the same here as it is in Australia, Africa, China, Russia, Italy, France, you name it. The scientific method is a universal methodology for acquiring scientific knowledge. And we know that science is the study of the universe. Alexa, define the scientific method.
The scientific method is an empirical method of acquiring knowledge that has characterized the development of science since at least the 17th century. It involves careful observation, applying rigorous skepticism about what is observed, given that cognitive assumptions can distort how one interprets the observation. Did that answer your question?
Yes.
Thanks for your feedback.
Observation is the first step of the scientific method. In order to have an observation, you have to have an observer, somebody who can do the observing. And in order to be able to observe, you have to have senses so that you can acquire external information. You can receive information through your skin. You can tell what temperature it is. Through your eyes, you can tell what the color of something is if you don't have color blindness. You can tell how far away something is. Depth perception, having functional eyes, gives you that sensory information. Ears, having functional ears, gives you sensory information about the audio vibrations in the fluid that surrounds you, right? Air, like sound waves. Taste, yum yum yum. Pizza, yum yum yum. Cupcake, ugh. I don't know, whatever. Cat food, ugh. Actually, if I was hungry enough, I'd probably eat cat food. Observation. It's how we know we are in a situation where we are receiving information, right? So, what do observations lead to? Observations lead to questions. Observations, you can have an observation that you create, that you have, that you make. You walk outside. You see a snake in front of you. You go, yeah! And the snake goes, whoa! And the snake goes. And you say, huh, why was that snake just sitting in the middle of the sidewalk? Why was that snake just sitting on that rock, sitting in the sun? Observation leads to questions, right? Now, questions lead to the next piece of the scientific method, which is a hypothetical thesis. A hypothetical thesis, a hypothesis. A hypothesis. A hypothetical thesis. A possible explanation to the question. Right? Okay? You've heard of that. Maybe. Maybe not. And now you have. Observations lead to questions. Questions lead to a hypothesis. Hypothesis. A hypothesis is an if-then statement. It's a testable explanation for an observation or a question. So if then. If blank, then blank. Now, some of you may have heard of the term independent variable. Some of you may have heard of the term dependent variable. I will make a list of vocabulary words, and I'll put them in the description of this video so you can just have them, and just go to something online on the internet, type in definition of independent variable. Watch some videos. Look at some images. Independent variable. What is the independent variable? Dependent variable. What's the dependent variable? I'm going to tell you, but don't just stop here. This is not where this ends. Okay? We are constantly learning. You're constantly getting better. You're constantly learning more. And if you stop learning, if you think you know, you're wrong. If you think you're done, you're not. If independent variable, then dependent variable. If IV, then DV. If independent variable, then dependent variable. We'll draw an explanation of this in a moment. Okay? So, that's your hypothesis. It's a testable explanation. After your hypothesis, what do you get? Well, we'll call this 3, we'll make that hypothesis. After your hypothesis, what do you have to do? You have to test it. You have to have an experience. You have to have an experience. Or which makes you an expert when you have enough experiences. You have to have an experiment. An experiment is an experience. An experiment is an experience. EXP. EXP, experience. EXP, experiment. Your experiment is the test that you do. It's a controlled experiment. Put in the word controlled as much as you can. Next. What do you get after your experiment? You get data. Your data, your results. Your qualitative and your quantitative data. Qualitative and quantitative. Qualitative is the color of something. Qualitative, the quality of it. Quantitative is a number. Oh, this is, I don't know, whatever. I don't know. Let me see. This cup weighs about, I don't know, it's 50 grams. I don't know. How do you do that? You get a scale. You get a ruler. You measure it. Okay? You take number data, quantitative, quantity, number. What's your quantity of bananas? I got 12 bananas. That's your quantitative data. Quantity, quantitative, quality, qualitative data. Okay? So, again, observation, question, hypothesis, experiment, data, then, what number am I on? I'm on 6. Analyze. Analyze the data. What does it say? What did the data tell you? What is the data? How many pieces of whatever do you have? Report the quantitative and qualitative data. Report your findings. Analyze the numbers. Look at the numbers, and then a conclusion is not an analysis. You are not writing your conclusion. You are just writing down your observations, your data observations of the experiment. So what you're doing is you're making an observation, you're forming a question, you're developing a hypothesis that is a potential answer to that question, then you're doing an experiment, which is going to result in more observations. But because it's controlled, your observations are going to tell you something. They're going to help you answer your hypothesis. Analysis, and then conclusion. And then your conclusion leads to more questions. Which brings you back. Because your conclusions are observations. Or your analysis and your data, those are observations. So your data, your analysis, and your conclusion are sort of clumped together in the general idea of data, of observations. It's your observations. Okay? That's a scientific method. Right? Okay? So let's look at an experiment real quick. So we make an observation, and we notice that plants grow better in in having white light. Okay? Having the full spectrum of the rainbow available to it. Okay? So when the plant has the full spectrum of the rainbow, it reflects green. Most plants. It reflects green, which means it doesn't absorb green. Which means it's absorbing the other colors. It's absorbing red, it's absorbing blue, it's absorbing orange, okay? It's absorbing yellow. The rainbow is being absorbed except for green. The plant is pushing green out. Ugh. Okay? So then we make that observation. Okay, plants are green. I wonder if certain types of light will affect plant growth. So, your hypothesis, if a plant is given red light, if a plant is given red light, then it will grow faster? Okay, sure, whatever. That's a hypothesis. Is this testable? Can I test this? Yes! Because I could just get some plants and give us some red light. Hey, let's do it! If IV, the independent variable, is the color of the light, independent variable, it's what I'm doing, what you are doing, what you do to the experiment, what you are doing to the thing you're testing, right? That's your independent variable, and the result is the dependent variable. Then it will grow faster. That's your dependent variable. Remember, hypothesis, if IV, then DV. All right? So, remember, this is a controlled experiment. So what we are going to do is we are going to attempt to replicate natural light in a laboratory. Okay? So we're going to have two plants. They're going to be the same type of plant. Okay? Now, listen, forgive my drawing. Also, forgive my spelling. I'm a phonetic speller, so I misspell things all the time. I'm wrong. All the time. All the time I'm wrong. So you have plants, two plants, same size pot, same amount of soil. Okay? Can I put a rose in this one and a clover in this one? No. They had to be the same type of plant. Okay? Same type of plant. Can one be a seed and one be, like a two-month-old plant? No. They got to be the same age. Right? They have to be the same height, roughly. Okay? Are you with me? Okay, good. So, we have two plants. Same size pot, same amount of soil, same species of plant. Can I give this plant, a gallon of water and this plant, 10 milliliters? No, I have to give them both the same amount of water, because what am I testing? I'm not testing how water affects plants. I'm testing how the color of the light affects plant growth. So, what else am I going to do? Am I going to put this one in a refrigerator and put this one in an oven? No, they're all going to be the same temperature. They're going to have the same temperature, right? Same temperature. Okay? Yeah. Now, what's going to be different? I'm going to have one plant receive light that is going to be similar to the natural spectrum of white light emitted from the sun. Okay, so this is going to be, as best as we can replicate it. Okay, if you wanted to, you could probably use natural light, but you would need to make sure that the timing of the light corresponds with the timing that the experimental light is going to the experimental group is going to get. I'm going to define that in a second. This one is going to get colored light. No tengo rojo. Okay? Now I have two groups. I have two groups. One group is receiving a different colored light. The other group is receiving natural light. Okay? Now, could I have this one receive light for 10 hours out of the 24 hours and have this one receive light 24 hours a day? No, they have to receive the same amount of light. So we'll just say 12 on, 12 off. 12 on, 12 off. Okay? The group that is receiving the general, the natural, the standard, the normal, the usual type of light is called your control group. You use the control group in order to tell the difference between a plant in standard conditions and how the plant in a varied condition, how they differ from each other. The group that receives the independent variable is your experimental group. Experimental group. Okay? So, this is it. That's all we did. Here we go. We have an experiment. What we do is we run the experiment. Say we run it for a month. 4 weeks. Okay? Every day we take measurements. Every day we get data. Okay? We water them every 3 days. Okay? Whatever. All right, we water them at the same time of day. Every time we water them. We water them with the same type of water. Okay? We don't dump out, that clean, fresh out of the plastic water, bloop, 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 and give it to one of them, and then go to the pond and scoop up water from the pond and give it to the other one. No. Okay? That's going to be, it's going to change the number of variables that you don't have control over in the experiment. You only want to have one variable that you're testing. Because if you test more than one variable, you don't know whether or not that variable had the desired effect and the other one didn't. Okay? Right. Yeah. That's it. So you get quantitative data by measuring the height, for example. You get quantitative data. You can at the end you can measure. You can do the masses. You can take them out of their soil and you can brush the soil off of the roots as much as you can. Rinse it off, right? So it's just plant. And then you get the mass of one, you get the mass of the other. See if it changed the mass of the plant, right? You would measure them every day, every 2 days, whatever, right? Then at the end, you look at the data and you look at how those differences, if any differences arise, okay? Then you analyze those differences and then you say at the end, yes, colored light does positively affect the growth of the plant. Or, no, colored light does not positively affect the growth of the plant. Or is it just the red light that doesn't affect the growth of the plant? Let's try blue. Then you do it again with blue light. Right? So, that's it. The scientific method and experimental design is universal within science, within the shared scientific community. Because remember, science is the study of the universe. And the universe is vast and ever expanding. We cannot study the universe in an individual lifetime. So what we do is with science is we break it down into pieces, little chunks that we all then can build upon for the future. Science builds the future. Okay? So, that's scientific method and experimental design. Next, we will be talking about, we'll be jumping directly into the beginning parts of biology. We are going to dip our feet into the stairs of the pool. Just go on in, and then eventually what we'll do is we'll be diving into the deep end, picking up a penny that you found. Okay. That is all. Stay strong. Stay true. You'll see me next time.